Well, hi everybody. Welcome to your final lecture of cell and molecular biology. Can't believe it. It seems like we just started a little while ago, but this is the very last lecture. This is the end of the material for Unit 3 as well as the end of the material for the final. So we'll come back to that idea at the very end, but again, this is the last lecture, Lecture 20. We're going to focus exclusively today on cancer. This is the second half of Chapter 20. Chapter 20 is a chapter on cellular communities, but this lecture is devoted entirely to the disease cancer. And before I go any further into the lecture, a couple things I do want to say about cancer. So, first of all, cancer is a disease of the cell. It affects the organism because the organism is made up of cells, of course, but cancer is a cellular disease. It's a perfect disease to highlight some of the major concepts that we've been covering all semester, or at least the second half of the semester, with regards to cell biology. And so not only is it of personal interest to many of us, but it's also this very enlightening vehicle by which we can describe some of these cell biology processes. The reason why I say that now is because you'll see some enthusiasm in my voice as we go through this lecture. There are aspects of cancer that are fascinating, that are intellectually engaging, and please don't ever take the wonderment or amazement of my voice for some lack of appreciation for how horrible a disease cancer is. I'm sure cancer has affected many of you personally. I will confess that it affected me very personally as well. My father's a cancer survivor, and my grandparents, who are no longer alive, are also victims of cancer. So don't ever think for a moment I don't have an appreciation or respect for this disease. I do, and please understand that I fully acknowledge and personally feel how horrible this disease is. But seen through the scientific lens, it is no less fascinating due to its horribleness. So let's just always keep that in mind as I go through this. So what are we going to be talking about today? Well, we're going to talk exclusively about cancer. We'll start off just by broadly discussing what cancer cells do. And from there, we'll begin talking about how cancer develops, or it starts in the patient. We'll talk about some of the triggers for cancer, what brings cancer on, both environmentally as well as genetically. We'll talk about the progression or persistence of cancer. And then we'll move on to the genetics of cancer. Are there such things as cancer genes? What does it mean to have a predisposition genetically to cancer? Near the end of the lecture, we'll talk a little bit about colorectal cancer, just as a specific example, get away from the broad themes for just a touch, and then we'll end the lecture talking about possible and future treatments for cancer, what is on the horizon. So what makes cancer unique among most all other diseases that we encounter as a species, as an organism, is that cancer cells are you. Most diseases that we think of are caused by outside invaders, by pathogens, things like the flu, long-term diseases, meningitis, and hepatitis, and things like this. These are outside pathogens that take over and try to invade our body. That's not the case for cancer. These outside agents are disease-causing agents. They can be other cells, viruses, bacteria, etc. And what marks these is that they're different from you. These are outside cells invading you, and they hurt you when they do so. Because these are outside invading cells and different than you, these pathogens can be fought. We talked about this very briefly earlier in the semester when we discussed antibiotics, but it's a very simple principle. Find something that the pathogen cell needs to survive that you don't. Find some molecular difference between your cells and the pathogenic cells, and then exploit that difference. Take that thing away. For bacteria, some antibiotics attack the cell wall. Our cells don't have cell walls. So if we disrupt the physiology, if we disrupt the chemistry of making cell walls, we disrupt bacterial cells and not our own. We exploit that difference. The pathogen will then die because you've taken away something that's required for its survival. You won't die because you never needed that thing in the first place, and the disease is cured with very little side effects to the patient. But the problem with cancer is that it is you, and that's the crux of the problem in treating cancer. How do we fight your cells, the cancer cells that are from you, without killing your other cells, the healthy cells that you need to continue to survive? In other words, what makes your cells that gave rise to the cancer different from your cells, which are healthy? Asked either in another way, what do your cancer cells need to survive that your other cells don't? What can we deprive the cancer cells of with a treatment and leave your other cells intact and side effect free? And the answer is there isn't much, and that's the problem. It's very hard to distinguish your cancerous cells from your non-cancerous cells, and so these treatments tend to be difficult. So the difficulty in fighting cancer, as well as the severity of the side effects, is in large part, if not exclusively, due to the fact that cancer cells are the same as the patient's cells themselves, and most things that are going to cause cancer cells to die cause healthy patient cells to die as well, giving rise to those wicked side effects. Now, what brings cancer on? Why is cancer even a disease of humans? Well, cancer is a blessing and a curse, or it comes from a physiological process that is a blessing and a curse. Our bodies can repair and renew. We know that. Between the cell cycle, giving rise to new cells, new daughter cells, and cell division, and stem cells, this kind of pool of pluripotent cells that can be many different cell types, many different differentiated cell types, we as individual human beings far outlive the vast majority of our cells. There are very few cells in our body when we die that were there when we were born. The vast majority of our cells that are in us when we pass away were not with us when we were born. These are new replenished cells that divided somewhere along the way as we grew into adulthood and beyond. They came from stem cells that we had in us as that pool or source of cells, but we as entities, as beings, far outlive most of our cells. Without these repair and renewal cellular mechanisms, our lifetimes would be much, much shorter. We would die when, theoretically, the last of our original cells died. What this implies is that we need a pool of dividing cells, of non-differentiated cells, that can replenish and renew our individual cells as those cells die, keeping our life going longer. But there's a price to be paid for that luxury of an extended life. In order for this replenishment and renewal system to work, we have to have cells in our bodies that are undifferentiated and poised to divide, poised to become proliferative cells, cells that are dividing. These are our stem cells. We have these cells' cellular behavior finely balanced on a fulcrum or a scale of being blocked from dividing when they're not needed, but allowed to divide when they are. This fulcrum or scale is balanced by cellular signals and signaling molecules, the regulation of genes for proteins that are needed for cellular division, as well as phosphorylation events to keep those proteins on or off. So we have a whole pool of proteins in our cells that promote cell division. We want those proteins somehow inactivated, either through cellular signaling, gene regulation, or phosphorylation regulation. We want them inactivated so that they're not driving cell division when we don't want it, but then we want to be able to easily turn those proteins on to allow the cell cycle to go on when those cells are needed to divide. So we have this pool of, I always think of these as angry dogs on a leash. We have these pools of cells that are dying to divide and want to divide, and they're pulling on that leash. Let me go, let me divide, let me into the cell cycle. But the cell holds them back with all its strength. And, like an angry dog on a leash, it doesn't take many mistakes. It doesn't take much for that dog to get away. It doesn't take much for that cell to get away and start blowing through the cell cycle. So when things go wrong, especially with these cells, they often go wrong catastrophically. Cancer is the example of this catastrophic breakdown. Cancer is a major cause of death on this planet. There was a CNN article very recently, I'm making this recording in 2014, saying that by 2018, it's projected that cancer will be the #1 cause of death in the United States. The majority of people who die in this country will be dying of cancer in 4 years from now. In Europe and North America, which includes us, 1 in 4 of us will die of cancer. 25% of us now, without those projections, will pass away of cancer. Cancer is, at its essence, a disease of aberrant cellular behavior and a breakdown of cellular rules. That's it. And I want you to open your mind a little bit, and I'm going to speak what sounds like analogies, but which is actually factually correct. We have to think of cells a little bit differently than we've ever thought of them before. Cells are social creatures. They are social things, just like us. We function in a society of many, many, many different human beings, and cells function in a society as well, a society of other cells. Cells live in large multicellular societies with a constant flow of information and molecule transfer. Cancer is a violation of the cellular social order, just as psychopathic or sociopathic behavior is a violation of our own societal order as human beings. Cancer cells are sociopathic. By definition, cancer cells are selfish, self-serving, and asocial. They have dropped out of the social norms of the multicellular community. In many ways, cancer biologists, to understand these processes, must be expert not only in cell biology, but other fields of biology as well, drawing knowledge from many different areas of the field. This is my own personal opinion, but I believe cancer biologists to be the most diversified and most multidisciplinary biologists in our field, because cancer touches all aspects of human biology. And ironically, much of what we know about cell and molecular biology, indeed much of what I've told you previously in this course, comes from experimental research into cancer. We understand how these processes in our cells should work by studying how they break down and don't work in cancer cells. That was a pretty long intro into the lecture, but what will we discuss today in this lecture? Well, we're going to talk about the causes and mechanisms of cancer, these types of cellular behaviors, these asocial behaviors that allow cancer to progress in a patient, and possible ways in which we can use this information currently to fight cancer, and maybe one day fight it so effectively that we have a chance of beating this horrible disease. So there are three main characteristics that define cancer cells. They are proliferative, which means they divide uncontrollably. They are invasive, which means they can get into other tissues and among other cells where they don't belong. And they can metastasize, which means they can spread from one organ system to another. A single living organism, multicellular organism such as ourselves, is, I hope you can appreciate at this point in the course, a highly complicated, highly coordinated, highly regulated system. Cells are constantly communicating with one another, and each cell should be doing what it does for the greater good. In other words, you can think of a single lung cell as being quite bored in its life. All it does is take in oxygen and pass it to capillaries, take carbon dioxide up from capillaries and release it back into the lung. Perhaps that lung cell desperately wants to be a muscle cell, or loves the pizzazz and the excitement of a neuron, but it can't be. It is doing its grunt work, churning out its activities, being its daily cell for the greater good of the larger organism. Every single cell in your body is sacrificing itself in many ways for you, for the greater good of the larger organism. And what makes this possible is cellular communication and signal transduction, what we've talked a few lectures ago about. This allows the coordination between these cells, and these cells can communicate with one another and keep each other on top of what's going on. So cells, by hearing signals from other cells at other parts of the organism, are constantly staying abreast and on top of what's changing in the body, what's needed in the body. And a single particular cell can respond to those changes and needs and contribute to the larger organism as best it can, given its role in the body. A cell, in the same line of thinking, should divide and enter the cell cycle when new cells of its type are needed. In other words, a skin cell should begin dividing when it's time for new skin to be made, or the previous layer of skin has been shed. It shouldn't divide any other time, and it shouldn't not divide when more of itself is needed. Again, sacrificing itself for the greater good. When does the skin cell divide? When it wants to? No. When more skin by the organism is needed is when that skin cell should divide. And when more of those cells are not needed, of course that cell should not divide. We don't need more of it. Same comes to cell life. A cell should live for as long as its usefulness is needed, and that cell should undergo apoptosis once the need for it has passed. Now, you've heard of apoptosis before, I'm sure, pre-programmed cell death or cell suicide. But think about this for a moment. It is every single cell's driving evolutionary force to survive. Yet we talk about apoptosis as a system for pre-programmed cell death. You send a molecular cue to a cell, and that cell kills itself. It flies in the face of everything evolutionarily. No cell should actively kill itself. Cells are fighting to survive. How and why does something like apoptosis exist? Cells are sacrificing themselves for the greater good of the organism. A cell will commit suicide when it's in the organism's best interest for that cell to commit suicide. The cell is essentially sacrificing its own life for the greater good and the greater life, the greater survivability of the larger organism. That is a very social behavior, not a selfish behavior. Cells need to maintain their cellular and biochemical characteristics despite all that might be going on around them. In other words, a lung cell should always behave like a lung cell and always make lung cell proteins. A cell should stay where it's supposed to stay. Lung cells should be in the lung. Essentially, cells have no free will. Cells are ants in the colony, each with its own job, each with its own role, and the colony will break down if ants stray from those roles, and the same is true of the larger organism. The larger organism will break down if each cell begins to exert free will. Cancer cells break all of those rules. In huge organisms like us, made up of literally trillions of cells, a single cell behaving asocially and aberrantly, a single cell that's breaking the rules is not a big deal. Just like a single individual in the city who is sociopathic is not a big deal. They can be corralled, they can be caught, they can be imprisoned or treated. It's not going to really cause the entire city to break down. But if this aberrant, asocial cell that breaks all the rules should happen to also become proliferative, enter the cell cycle, begin dividing, when it divides, it makes new daughter cells that have the same exact asocial characteristics as itself, the same cellular instability as itself. That becomes a problem. It's like all the sociopaths in the city begin having children who are sociopaths, and those children have children who are sociopaths, and the population of sociopaths grows, and it grows faster than the normal population because the sociopaths are dividing and reproducing even more rapidly. That could easily lead to the breakdown of the city. So what happens in this situation for cancer cells is you get a mass, a population, a tumor, of rapidly growing cells that are not contributing to the entire whole of the organism. They are not sacrificing themselves for the greater good. Instead, they are quite selfish. And this large mass of rapidly dividing aberrant cells crowd and push into and destroy the healthy tissues that happen to be around them. That's cancer. The textbook defines cancer as a relentlessly expanding clone of abnormal cells. What a perfect 6-word description of this disease. That's all it is. Relentlessly growing tumor of asocial cells. Cancer is you. Cancer is your own cells no longer giving a damn about the larger organism, no longer willing to sacrifice themselves for the greater good, but instead doing what cells have done on this planet for millions of years, putting their own cellular survival as the first priority, and having the survival of the larger organism secondary. Sound familiar? Something that continues to grow and divide and multiply and invade and push into its surroundings, no longer giving a damn about the greater good, no longer giving a damn about the healthy populations of life around it. Cancer cells are defined by 2 main properties, which they pass down to their daughter cells. They proliferate like crazy. There's no human cell that divides more rapidly than a cancer cell, with the exception of embryonic cells during development. And they do this in defiance of normal constraints. Most cells, when they start growing or dividing, will respond to cellular signals, and cellular signals can shut down the cell cycle, cause those cells to enter G0. Cancer cells do not listen to those signals. They defy those normal molecular constraints. Cancer cells are invasive. They colonize cellular tissue regions that are reserved for other tissue or cell types. It's these 2 properties, when they're taken together, combined, that makes cancer so utterly devastating. These properties, they grow rapidly wherever the hell they want. That's cancer. It's not just a lung cell growing rapidly in the lung. It's a lung cell growing rapidly in the lung that then pushes into the diaphragm, that then pushes into the circulatory system, or spreads even more. Cells with the first property, dividing like crazy, but not the second, they're not invasive, give rise to what we refer to as a benign tumor. First thing you ask, you hear someone, there's a lump found, there's a tumor found. Was it benign or malignant? And it was benign. Oh, thank God, thank goodness. We understand that benign means not harmful or much easier to treat, but this is why. A rapidly growing mass of cells that remains localized and non-invasive, that stays exactly where those cells should have stayed, can usually be isolated, effectively removed by surgery. These are benign tumors. When the answer is, was it benign? No, it was malignant. Oh, no, it was malignant. Now we know we're talking about cancer. Malignancy implies the invasiveness of cancer. A tumor can only be classified as malignant if it possesses the capability of invading surrounding tissues. So it's dividing like crazy is bad, but dividing like crazy alone can be treated. Dividing like crazy and invading, that is cancer. And so to highlight the differences here, we have a schematic of epithelial cells, skin cells, normal cells on the top. See the benign cells, the benign tumor? It's dividing like crazy. These cells aren't really clustering the way they're supposed to. They're no longer behaving appropriately, but they are confined, and they are not invading the healthy cells around them. And so this benign tumor can easily be cut out. Malignant cancers are not constrained. They are not isolated. You can see these cells have invaded the neighboring healthy cells. How do you easily remove this tumor? And obviously you can see even in the schematic you can't, and that's what makes cancer a challenge. The last and most frightening classification of cancer is the metastatic cancer, the most dangerous. This is where the tumor is not only invasive of cells in its neighboring population, but these tumor cells have migrated to or invaded the circulatory systems, either the blood system or the lymph system. And these circulatory systems take those cancer cells to other parts of the body. Once a cancer has gone metastatic, it can theoretically spread anywhere. The blood or lymph carries those cancer cells to other parts of the body. Those cancer cells will eventually come out of the blood or lymph. They will land and adhere to some other tissue type, and they will start being cancerous again. They will do what cancer cells do, divide like crazy and invade. When they divide like crazy and invade in these new places, they give rise to new tumors in those areas. Those are secondary tumors. Secondary tumors are common in metastatic cancers. Here we see a liver riddled with secondary tumors. Interestingly, these secondary tumors retain many of the properties of the primary tumor. They are as invasive and as rapidly dividing as the primary tumor, that's one. But more importantly, and perhaps more interestingly, is you can tell where the primary tumor was by studying the secondary tumor. In other words, a secondary tumor in the liver, if it came from the lung cell, you could isolate those cancer cells and they will look physiologically like lung cells. They will have all of the molecular and physiological hallmarks of lung cells because they came from lung. Yet you're finding them in the liver. You will instantly know as the cancer biologist that these are secondary tumors of the liver that had come from or were given rise from primary tumors in the lung. Once the cancer spreads in this way, metastatically, it becomes much, much harder to eradicate. So let's talk a little bit about epidemiology. Epidemiology is the study of statistics, using statistics and large-scale analyses of populations of patients or people to analyze the relationship between certain environmental factors and human disease. So if you're interested in seeing why is it that tuberculosis is spreading mainly in subway riders of New York City, epidemiology is the field that you are discussing there. You are analyzing, using large sample sets and statistics, the relationships between the disease onset and subway ridership, for example. So we can do these types of studies with cancer. And if by doing so, we can link certain environmental factors to cancer, and we can remove or minimize our own exposure to those cancer-causing factors, then we've gone a long way towards, well, even doing better than curing cancer. We've gone a long way to avoiding cancer altogether. And indeed, the environment is almost always a contributing cause for most types of cancer, although most of the time it's not solely responsible for causing cancer. Let's look at some empirical evidence for the epidemiology of cancer. The types of cancer and the incidences of cancer are usually regional, not demographic, not by ethnic background, nor familial. This shows that strikingly, this is the cancer mortality rates for colon cancer among white males for almost 25-year period by state economic area. And you can see the more red the number, the higher the incidences of cancer, the more blue the number, the lower the incidence of cancer. And is it family? No. Is it ethnic background? No. What seems to be the main contributing correlation between colon cancer and populations of people who have it in the United States? Region. Look, most people in the northeast, or the highest rates of cancer, are clustered in the northeast of the country. That is an environmental effect, not genetic. Migrants, immigrants, people who come from other areas will more often get the cancer of their new home, not their old one, again implying an environmental effect. In other words, if, for argument's sake, prostate cancer levels are very, very high in Mexico, but colon cancer levels are very, very high in New York, a Mexican immigrant who is born in Mexico, of Mexican descent, nothing but Mexican genetics, who emigrates to New York, will more likely get colon cancer as an older adult than prostate cancer. They will get the cancer of their environmental home, not the cancer of their genetic home. Epidemiology first showed that cervical cancer was likely to be a sexually transmitted disease. You probably know this, but cervical cancer is caused by human papillomavirus, or HPV, and it's much, much more common in sexually active women who have had multiple partners. So this is definitely an environmental effect. There's a vaccine for HPV available. I'll reserve my personal judgment on that. But again, this vaccine has had quite a bit of light shed on it, both positive and negative, in recent years. But please know that things like viruses causing cancer is the rarest of rare exceptions. The vast, vast, vast majority of cancer is not transmissible. The vast majority of cancer is a spontaneous event within the patient. Some more environmental effects. Tobacco. Of course, we must discuss tobacco in this conversation. Tobacco is indisputably the leading environmental cause of cancer on this planet. Virtually every single case of lung cancer can be traced back to tobacco, directly or indirectly. Smoking also substantially raises an individual's risk of bladder cancer. If we were to make smoking cigarettes illegal tomorrow, ban it from the entire population, it's estimated that we would prevent about 30% of all cancer-related deaths in this country. That is an incredible statistic. Almost a third of all cancer-related deaths would not happen if we made smoking cigarettes impossible. There is no other single policy, law, action, or medical treatment that could ever have hope of shifting cancer-related deaths as dramatically as that. Isn't that amazing? And yet we continue to do it. We continue to retain the right to slowly allow ourselves to kill ourselves. And perhaps that's a good thing. It is a free country. Perhaps it's not. I'm not judging anybody. I'm a former smoker myself. I smoked for a number of years. So I'm not judging anybody's personal choices, but obviously know the severe impact that smoking has on your health. This is for men, so it's not entirely appropriate to vape path, but still it shows the difference. It's not so different in women, I'm sure. This is the relative risk of lung cancer, according to the duration and intensity of smoking in men. And this is the years of smoking down here, and this is the relative risk. So you smoke less than 10 cigarettes a day for less than 20 years, and your risk has gone up above zero, but not appreciably. Look what happens after the 45-year mark. You smoke for 45 years, 10 or less cigarettes a day, half a pack a day or less. Still, your risk has gone up considerably, but not dramatically. If you smoke 20 cigarettes, a pack a day or more, it's gone up quite a bit. Once you pass that 50-year threshold, look at this. Look at this relative risk. Your relative risk has gone up almost 50%. If you smoke more than 20 cigarettes a day for 50 years or more. That's an incredible increase in relative risk. There's no other environmental effect that can come close. Remember that most lung cancer deaths, if not all lung cancer deaths, can be traced back to tobacco abuse directly or indirectly. To put that in some perspective, look at the estimated cancer deaths by where those cancers develop in 2012. All of the other cancers that we think of as common, colon cancer, breast cancer, pancreas cancer, or pancreatic cancer, prostate cancer, these are the cancers many of us are aware of, many of us have been touched by. Again, my father is a prostate cancer survivor. All of these cancers taken together sum up all of these cancer deaths in 2012, and still they do not match the number of deaths due to lung cancer alone, and most of those lung cancer deaths are due to tobacco abuse. Amazing. Amazing. Obesity also significantly raises the overall risk for cancer long term. It's unclear how. We are well aware that obesity, prolonged obesity, increases risk of late onset diabetes, heart disease, but cancer risk also goes up. The mechanism by which is still not clear. So, very much so, lifestyle choices and decisions that you make, even at this point in your life as a young adult, can greatly enhance or hurt your overall quality of life moving forward. So let's talk about how cancer forms genetically then. That's the environmental impact or effects on cancer, but how is cancer developing genetically at the DNA level? I said it already, but it's important to point out that cancer is not a disease solely of the environment. It is a nature-nurture disease. It's a disease of cellular mutation in the end, and mutations are changes in the DNA. Anything that increases the mutation rate in a cell can and does increase the overall risk of cancer, and this includes smoking. We will never completely eradicate cancer, even if we did outlaw cigarettes and everything else that we can think of, because we know that mutations occur spontaneously. We discussed that in the DNA mutations lecture in this course. There are spontaneous mutations, and mutations cause cancer, so there will always be spontaneous incidences of cancer. So fundamentally, at its core, cancer is a disease that is born from inside of us. What makes cancer so unique as a genetic disease is that it is exclusively a somatic mutation disease, never germline. Hopefully you remember these terms from the lecture on DNA mutations. Somatic mutations are mutations that affect populations of cells in the adult. Germline mutations occur in the gametes. Since germline mutations do occur in the gametes, they affect the entire organism. If there were to be a cancerous mutation in a germline cell, the entire resulting organism in the next generation would be cancerous, an entirely cancerous embryo. Does that happen? Yes, it does. There is such a thing. What is the result? Of course, such an organism is not a human being. A cluster of cancerous developing cells in a uterus is not a person, is not a human being, and so that organism is not viable and would not survive to birth. So cancer is somatic. We already have an appreciation for the amazing properties by which DNA can fix itself while it repairs and replicates, but our cells are replicating a lot, and there's a lot of DNA in those replicating cells, and that DNA is replicating in those cells a lot of the time. We've got trillions of cells replicating billions of bases of DNA often. We talked about the error rate of DNA replication a while back. We said that one mistake slips by for every 1 to 10 billion DNA bases copied. That's the spontaneous rate of DNA polymerase mutation or mistake. This was the mutations that were fixed by DNA mismatch repair. This is with no environmental effect at all. This is the cell humming along optimally. Is that an awesome error rate? It's unbelievable. It's one hell of an impressive accuracy rate. There's nothing known that is more accurate than DNA polymerase. But mistakes slip through. To be pessimistic, one mistake slips through. Every 1 to 10 billion DNA bases, yeah, but one mistake slips through. And that mistake could lead to disease. If you do the math, you do the estimates, we have so much DNA in our bodies and so many cells in our bodies, even with this phenomenally low error rate, a single human being has roughly 10 to the 16th cell divisions occur in his or her body in a single lifetime. That is a 1 followed by 16 zeros. If you take into account the error rate of the DNA polymerase, that means statistically that every single gene in your body, every single gene in your genome, will undergo about 1 billion separate mutations somewhere in your cell over the course of your life. Every gene will be subjected to about 1 billion mutations over the course of your life. When looked at that way, it's amazing that we don't all develop cancer much earlier in life with so many mutations. What protects us from early onset cancer, if we have so many mutations in our genes? What protects us is the randomness of these mutations. In fact, not to diverge too much here, take a tangent, but what protects us also is junk DNA. A lot of those mutations are occurring in junk DNA. One major theory for why junk DNA exists and persists evolutionarily is it is a place for DNA to be mutated that kind of draws those mutations away from important genes. But what protects us from early onset cancer from these mutations is the randomness of these mutations. Cancer rarely results from a single mutation in a single gene. Instead, cancer is caused by an accumulation of very specific mutations which come together in a very unique and specific combination that allows cancer to progress. Remember, at its simplest consideration, in order to be cancer, a cancerous cell must be proliferative and invasive. It must divide uncontrollably and invade neighboring tissues. Those are two completely different properties that cancer cells need. This requires mutations in the cell cycle checkpoints for the proliferative activities and mutations in the cell adhesion pathways that allows the invasiveness to occur. So right there, even at its simplest consideration, two separate cellular processes must be mutated, cell cycle and cell adhesion, in order for cancer to be possible. And it is these rare specific mutations that are needed for cancer, therefore, it becomes rare for these specific mutations to occur together in a single cell. The randomness of mutations mean that these specific combinations of mutations rarely come together in a single cell, and so a single cell rarely mutates to become cancer. Unless you've given enough time for those mutations to accumulate. If a sufficient amount of time has passed, and a sufficient amount of combinations of mutations has occurred, then you begin to get the possibility for cancer to occur. It's like saying, well, having a cell go cancerous is like playing the lottery. It's very rare and very unlikely to get that specific combination of numbers together on a lottery ticket to win. So if you play the lottery once, do you win? No. If you play it 10 times, do you win? No. But your cells are playing this lottery every single day for 75, 80, 85 years. If you were to play the lottery thousands of times a day, every day of your life until you were 80, you would very well expect to win at some point. And the winning ticket here is the accumulation of mutations that causes that cell to go cancerous. It takes a long time to play that many lottery games, but eventually that combination can and will be obtained. This, more than any other reason, is why cancer tends to be a disease of old age. Because with that passage of time, you've had much more opportunity to accumulate those mutations. Does cancer occur in young people? It does. Those people had that combination occur much earlier in life. It's like winning a lottery you don't want to win much sooner in life with much fewer games played. But most people have to continue drawing from much, much later in life to allow those mutations to accumulate. And so cancer tends to occur later on in life. In fact, usually even a whole bunch of mutations together in the same combination isn't enough to cause cancer. We need even more. We need what is referred to as genetic instability. Genetic instability is a specific set of mutations which allow more mutations to occur more readily. And it is often seen to be causally related to cancer progression. When mutations themselves affect the accurate replication and repair of the genome, we see large-scale genetic instability. We see large-scale chromosomal mutations occur. This is the biggest cancer risk. So what types of mutations do this? Well, if you have a mutation that deactivates your DNA repair enzymes or your DNA proofreading capability of your polymerase, what would you expect to happen? If the DNA repair enzymes or DNA proofreading is inactivated, you're going to get more mutations. It's like playing even more lottery games. you're going to get even more mutations and even more combinations much more readily and rapidly than you would have before. You're increasing your risk of cancer. Other examples of this, bypassing the cell cycle checkpoints. Remember P53? P53 was the cell cycle checkpoint protein that caused the cell to stop dividing when DNA mutations were sensed. If we have mutations in P53, cells will not arrest in the cell cycle when there are mutations. They will instead continue on dividing and copy those mutations onto the next generation. This increases the error rate. Increasing the error rate is playing more lottery games. That increases your risk of cancer. Mutations that cause defects in the mitosis machinery cause genetic instability on a much larger level. Now we get the wrong number of chromosomes going into the wrong cells. We have large-scale chromosomal mutations. These types of simple, small-scale mutations on the DNA repair enzymes, the cell cycle checkpoint proteins, the cellular machinery needed for mitosis, these cause genetic instability. These mutations cause greater, larger-scale mutations in the cell, which can directly lead to and result in much larger, more severe accumulated mutations, eventually perhaps leading to cancer. The entire genome can be radically altered in this way. And we see examples of that. This is a karyotype. A karyotype is a collection of human chromosomes that have been stained in a particular pattern. The karyotype in the unstained is here on the left, and then we have these fluorescent stains on the right. The fluorescent stains are created so that each chromosome has its own color. So each chromosome should be its own color, as we see here. Pink, orange, light blue. All of these chromosomes that you see that are multicolored, these are the results of translocation events. These are the results of chromosomal swapping. This purple piece is not supposed to be here. This is from another chromosome. This green piece is from another chromosome. This pink piece from another chromosome. It's hard to tell, but there's even a purple piece here that doesn't belong. This chromosome here is a chimera of 3 separate chromosomes. This chromosome is made up of 2 separate chromosomes. 2 separate chromosomes here and here and here. 3 separate chromosomes here. This is chromosomal instability. These chromosomes in this individual have essentially fractured and glued themselves together randomly in any conceivable pattern just to have chromosomes intact again. These are chromosomes from a cancerous cell, and this cancerous cell is under extreme chromosomal instability. So if you take a moment and think about it, rapidly dividing cells have the potential for a more rapid evolution, and this is the problem with cancer. All cells on this planet are fighting to survive. It's just how they do so that is different. Individual cells fight to survive on an individual basis. Cells that are part of a larger multicellular community fight to survive by keeping the organism alive. But mutations that we are discussing here should not be mistaken as crippling mutations. Cancer cells are bad for us, but selfishly, asocially, cancer cells are great for themselves. In fact, cancer mutations make cancer cells supercells, and that is the danger of cancer. Cancer cells survive better. They are invasive. They are outcompeting. They can overtake and outperform normal, healthy cells surrounding them. Evolution likes this. Natural selection favors this. Natural selection awards the cells that survive the best, period. Natural selection doesn't give a damn about the health of the organism. Cells that divide the fastest, survive the best, win by surviving into the next generation. Couple this natural selection bias with cancer's rapid cell division, and evolution begins to work against the patient. Some properties that make cancer a supercell are the rapid division, of course. It's very easy to outcompete your neighbors when you're dividing and growing faster than them. But its invasiveness also. You get into your neighbor's area. You steal your neighbor's food supply. Cancer cells have the ability often to attract their own blood supply. Cancer cells can cause new blood vessels to grow towards the tumor, providing the tumor with the needed food that it needs to continue to grow. Cancer cells can degrade the extracellular matrix, speeding their growth and their invasiveness. And, as we've already said, cancer cells can move, they can migrate, and enter the bloodstream or lymph system, carrying those cancer cells to new locations. These are the more obvious, already discussed properties of cancers as a supercell. What else can cancer cells do that give them an advantage? Cancer cells are deaf to the larger cellular signals that are passing around in the body. Cancer cells no longer require signals from other cells to thrive. All of the cells in your body that are healthy right now are alive because they're getting signals from other cells saying, we need you. Don't go into apoptosis. Don't kill yourself. We need you. Without those signals, your cells would undergo apoptosis and die. One of the main proteins that plays a role in this is RAS. We talked about RAS very quickly in the end portion of our cellular communication lecture. RAS is turned on by receptor tyrosine kinases. An example of a mutation that causes cancer cells to thrive in the absence of cellular signals is a mutation that we often see in RAS. This mutation in RAS that promotes cancer locks RAS in its active state. You might remember that RAS is a G protein-like protein. It binds to GTP when it's active and GDP when it's inactive. Here's the receptor tyrosine kinase. It phosphorylated itself. This is one of those accessory proteins, and the accessory protein eventually activates RAS. RAS, like a G protein, is on when it's bound to GTP and off when it bounds to GDP. This mutation in RAS keeps it on even when GDP is bound. This mutation fools the cell into thinking that it's continuing to get a signal through its receptor tyrosine kinase telling it to divide. This signal binding to this receptor is telling this cell, keep dividing, we need more of you. Keep dividing, we need more of you. The receptor is activated through autophosphorylation, RAS is turned on as a result of that, and RAS sends that signal deeper into the cell. Keep dividing, they need us. Keep dividing, they need more of us. This mutation in RAS keeps RAS on. It keeps RAS on independently of any other signal. The signal, we need you, keep dividing, ceases. The body doesn't need any more of this cell. The receptor tyrosine kinase deactivates and is no longer phosphorylated. The accessory proteins are no longer recruited, and RAS should be off. But RAS is mutated, and it's on. RAS, in its active state, continues sending the signal deeper into the cell. Keep dividing, they need more of us. Keep dividing, they need more of us. But it's not the right signal. Going back to the analogy we used in the cellular communication lecture, remember, when we say the cable guy is coming tomorrow, the cable guy is coming tomorrow, and if we keep saying that into tomorrow, the signal is no longer accurate. We're no longer giving correct information. The textbook says this is like a faulty doorbell ringing when no one's there. The doorbell is broken. There's a short in the circuit. No one's pushing the button. You go and answer the door and there's no one there. The cell continues to divide, but more of it isn't needed. Another thing that cancer cells do, making that a supercell, they no longer default to apoptosis. Cancer cells are no longer willing to take the bullet for the larger organism. It's perhaps one of the most frightening characteristics of the disease. Normal cells will kill themselves in a moment to sacrifice themselves for the greater good of the organism, and normal cells will do so when they cease receiving the correct signals to survive. Not cancer cells. Again, they're asocial. They're selfish, and they're in it for themselves only. Cancer cells will continue to divide, regardless of what signals they're receiving or not. Cancer cells do not undergo apoptosis. Apoptosis was largely an effect of p53. DNA damage led to p53 activation. p53 activation halted the cell cycle. If the DNA couldn't be repaired in a given amount of time, the cells defaulted to apoptosis. If p53 is mutated, we don't stop dividing at the DNA damage. We don't have time to repair that DNA, and we certainly don't default into apoptosis. Cancer cells are immortal. They never stop dividing, while normal cells do. The cancer cell lines that were worked on in laboratories have been used since the 50s and 60s, and they continue to divide. It's amazing. Cancer cells are genetically unstable. They show genetic instability. They have large-scale chromosomal mutations. Cancer cells are abnormally invasive. This is partly due to a lack of the cadherin protein we just talked about earlier, and other proteins that are needed for the cell junction as well. Lack of adhesion means that these cells are no longer part of a larger tissue. Lack of adhesion makes it easier for the cells to slip out of that tissue and metastasize. And how were these adhesion proteins lost? What do we mean we lost cadherin? Gene mutations. Promoter mutation. You no longer transcribe the cadherin gene. Cancer cells can adhere to, survive at, and divide in foreign tissues of other cell types. This is also uniquely cancerous. This is the very description of metastasis. Normal cells, if you were to take a normal healthy lung cell and put it in the liver, it would die within days. When it's misplaced, it's getting the wrong cues, the wrong cellular messages from the neighboring cells. As soon as the wrong cellular cues are being received, as soon as the cell doesn't hear the language it's used to in its environment, it defaults to apoptosis, not cancer cells. Cancer cells are deaf to those signals and so continue to divide. So, if largely proteins are so important, let's get a closer look at these proteins' genes. All of these 6 points I've just referred to on these last 2 slides are governed by proteins. So let's get a look at the genes for those proteins. There are 2 very general classes of gene mutations that could potentially cause cancer. They are genes that should be off in healthy cells, but when they're mutated and become hyperactive, they cause cancer. We've referred to these genes as oncogenes, cancer-promoting genes. These are genes that are on when they should be off, or highly active when they should be slightly active, and it's this greater enhanced gene activity that causes cancer. Here, the protein activity is usually what's contributing to the cancer. If the gene is unmutated and responding healthily, if it's off when it should be off, we call it a proto-oncogene. In other words, it could be an oncogene, but it's not an oncogene now. And then there's the opposite effect. There are genes that should be on to help stop cancer from developing. But when these genes are mutated to become off, or more inactive, cancer is a risk. We call these tumor suppressor genes, because when these genes are on when they should be on, they suppress tumor formation. Here, the protein, when it's made, is protective against cancer. So we want the protein around, and when the mutation causes the protein to not be around, cancer is a risk. It's the absence or attenuation of the protein that gives the cancer a chance to take hold. Oncogene mutations are genetically dominant, while tumor suppressor gene mutations are genetically recessive. What we mean by that is to get an oncogene effect, you need only one oncogene allele. To get cancer from a tumor suppressor gene, you need 2 mutant alleles. And I want you to think about why that is. We'll come back to this in class, but I want you to think about why oncogenes are dominant and you only need 1 allele, while tumor suppressor genes are recessive and you need 2 mutant alleles for cancer to develop there. It's the recessive property of tumor suppressor genes that also explains the genetic predispositions to cancer we hear about sometimes. We often hear there's a genetic predisposition to breast cancer. She has the breast cancer gene, but she doesn't have the disease, so what should she do? Well, this is all regarding tumor suppressor genes. If you have 2 healthy, wild-type tumor suppressor gene alleles, you don't have cancer, because it's recessive. To develop cancer, you need to develop mutations in each of those 2 healthy alleles, because you need 2 mutant alleles for cancer to develop. The probability of this is the probability of mutating 1 allele times the probability of mutating the other allele. In other words, the probability of getting cancer when you have 2 healthy, non-mutated, wild-type tumor suppressor gene alleles is the probability of getting 1 mutation squared. Now imagine that you are heterozygotic. You have 1 healthy, functional tumor suppressor allele, and 1 of your alleles is mutated. You still don't have cancer, because it's a tumor suppressor gene. You need just 1 allele to be cancer-free. But to develop cancer, now you only need a mutation in your 1 good remaining allele. If you get a mutation in that 1 allele, and that 1 allele only, cancer will develop because the other allele was already mutated. The probability of getting cancer, in that case, is the probability of getting that 1 mutation in that 1 allele, period. Not squared, because the other allele was shot to begin with. This is the genetic predisposition of cancer. If I am heterozygotic for the tumor suppressor allele mutation, and you are homozygotic for healthy wild-type tumor suppressor gene alleles, my risk for cancer is much higher than yours. my risk for cancer was an inherited risk because I got that mutation from a parent. Thus, I have a genetic predisposition to cancer. It's easier for me to get cancer, and it's easier for me to get cancer because of my genetics. That is how we have a genetic predisposition for cancer. The BRCA gene works this way. And we've heard about the BRCA gene recently. Angelina Jolie got preventative mastectomies because she carries these alleles. Does she have cancer now? Did she have cancer when she had the mastectomies? No, because she had working alleles. But she had the mutant alleles too, so her risk for cancer was elevated. Carriers and non-carriers, these are the genetic terms for this. Hopefully you remember your genetics. A carrier is just another way of saying a heterozygote. Non-carriers are homozygotic wild type. Look at the risk for breast cancer in these two individuals. You can see that the risk of breast cancer in the heterozygotic carriers is much, much higher because they already have one mutant allele. For these individuals to develop breast cancer, they had to pick up mutations in 2 of those BRCA gene alleles independently, and that's a much harder thing to have happen. Proto-oncogenes and tumor suppressor genes can be lots of different things at their protein level. They can be growth factor proteins, receptor proteins, G proteins, DNA repair proteins, proteins that regulate the cell cycle, proteins that initiate apoptosis, cell adhesion proteins like cadherin, the list goes on and on. This figure represents all of the pathways that are known to contribute to cancer, with the proteins indicated in green being large-scale cancer players. We can see catinin here. We'll talk about that in just a second. We have the BCL and death factor proteins here. They promote apoptosis and cell death. We've got survival factors. We've got our receptor tyrosine kinases and RAS, which we've heard about before as well, the integrins of the adhesion pathway in the extracellular matrix. But also in red are other proteins of these same pathways that also contribute to cancer, lesser players. For the cancer cells themselves, it doesn't matter if an oncogene has been mutated to become active or a tumor suppressor gene has been mutated to become inactive. All that matters is that the physiology of the cell has been changed. The cancerous cells are now dividing when they shouldn't be. And for most tumors, both oncogenes and tumor suppressor genes have been mutated over time. Keep in mind that cancer is a disease of accumulated mutations. What has consistently become the more important parameter for cancer to develop is not the type of mutation. Is it an oncogene? Is it a tumor suppressor gene? Is it a point mutation? Is it a chromosomal mutation? Much more importantly is where that mutation is found. More specifically, in what pathway, what cellular pathway that mutation is found. What gene does that mutation affect? What does that protein that the mutant gene encode do? And what pathway is that protein part of? By pathway, we mean these stepwise processes that are mediated by proteins. This is glycolysis. Bad memory, I can hear you groaning from here, you former biochemistry students. Glycolysis is not a pathway that tends to be affected by cancer. Glycolysis is not cancer-forming, but it is a perfect example of a pathway. These circles represent intermediates along the way of the pathway, and proteins, enzymes, mediate each of these arrows. So the pathway is made up of proteins. Going back to this figure, each of these is a pathway as well. Pathways of proteins that carry out some cellular process. There's the survival factor pathway that goes from protein to protein to protein to protein, eventually causing changes in gene expression and changes in cellular proliferation, etc. So there are these protein pathways. Rapid and cheap DNA sequencing technology is now allowing us to sequence the genomes of different patients, more importantly, sequence the genomes of different tumors and compare the DNA mutations between those tumors to see what they have in common and what's different. By doing these tumor sequencing projects, we can track the unique mutations that cause the cancer in those tumors in a very individualized tumor-by-tumor way. And what we have found by doing this is that mutations causing cancer tend to fall into genes for proteins that really affect only 4 different pathways. One of those pathways is the cell proliferation pathway. Well, that was no surprise. Of course the cell proliferation pathway is going to be affected. It is the very defining characteristic of cancer. The control of cell growth is also affected, the response to DNA damage, and the response to cellular stress. The vast majority of mutations that accumulate in cancer tumors fall into pathways that affect these four, and only these four cellular processes. Pretty amazing. So alterations in cell proliferation, cell growth, response to DNA damage, and response to cellular stress are the promoting causes of cancer. The different single genes that are mutated in these different tumors are different. But what is common is that these pathways are affected in almost all human cancers. What mutations there are, what genes are affected, how the pathways are altered, differs between different patients and different tumors. But the fact that these four pathways are affected tends to be conserved. It is the under or overactivity of these pathways that is believed to cause cancer. Alright, so let's wrap up this lecture talking about a specific example, and then we'll just discuss some strategies for fighting cancer, and we'll be done not only for this lecture, but for this semester as well. Colorectal cancer is a cancer of the epithelial lining of the colon and or the rectum. It's typically associated with old age, so it's a disease of accumulated mutations, not necessarily genetic. But a small proportion of cases do exhibit a genetic predisposition, and we know what that means now. So this points to a tumor suppressor gene, because only tumor suppressor genes have the ability to be recessive, to give a genetic predisposition. In one documented family with this predisposition, the mutation was in the allele for a DNA repair enzyme, dovetailing with what we've just discussed. That's not a surprise to us anymore. In another family, the genetic predisposition of the cancer develops quite early in life. The cancer itself is preceded by the development of hundreds of thousands of small tumorous growths called polyps, which you may have heard of before. This is a polyp in the colon here. It's essentially a wart. It's a cluster of dividing cells that is not yet invasive or metastatic. These polyps are precancerous cells because they have one of the characteristics of a tumor. They are rapidly dividing. They are proliferative cells. That's a precancerous cell type. To be cancerous, it also needs to be invasive. This cell is not invasive yet. These polyps and the cancer that could follow from them are due in this family to a mutation of a gene called the Adenomatous Polypus coli gene, or the APC gene. Now, we've heard APC before. This has nothing at all to do with the anaphase promoting complex of the cell cycle. Do not confuse these two APCs. Predisposed individuals that are carriers for this mutation are recessive but heterozygotic for the mutation, of course. When the wild-type protecting tumor suppressor allele mutates, now we have two mutant alleles and the cancer can develop. So if mutant APC causes this cancer, what about everybody else who didn't have the genetic predisposition? What about all the people who just get colon cancer out of the blue? Those people who were born homozygotic wild type for the tumor suppressor gene. Well, believe it or not, in over 60% of those other cases of colorectal cancer, when we look and see what is going on in the genomes of those cancers, we see that both APC alleles are missing in the cancerous cells. So even though the family gave us a clue of the role APC was playing in this cancer, we now know that APC plays a similar role in most other colorectal cancers as well. Both alleles are present and functional in the healthy tissue. The APC alleles are missing in the colorectal cancer tissue. Other chromosomal mutations led to the loss of those alleles. You say, well, how do you lose two alleles, especially when you were born with two healthy ones? Other mutations cause those. These genetic instability mutations. Mutations in the mitotic machinery may have resulted in those chromosomes not going to a daughter cell. Mutations in the DNA repair machinery may have allowed an accumulated set of mutations to affect the APC genes. Chromosomal instability might have resulted in deletions of those chromosomal regions containing those alleles. In any event, it's the loss of the APC alleles that causes the cancer. What causes the loss of the APC alleles themselves are further mutations that occurred earlier in the cell, leading to that genetic instability. What is APC? APC is an inhibitory protein. It blocks a cellular signaling pathway that causes cell proliferation to occur. Normally what happens in a cell is that there's a signal that promotes cell growth called Wnt. Without the Wnt signal binding to its receptor, we have an inactive cellular signaling protein. We don't carry the signal into the cell. APC is active in the absence of this signal, and it destroys a protein that's responsible for cellular proliferation, and so those cellular proliferation genes are off and the cell doesn't divide. Normally, in healthy tissue, when Wnt is present, Wnt binds to its receptor, this activates the second messenger turns off APC. Without the destruction of this yellow protein, these genes are turned on, and these genes encode proteins that cause the cell to divide. So with the Wnt system running, these cells are proliferative. Without the Wnt system, APC keeps these cells from dividing. What happens when you lose the APC alleles? When the APC alleles are lost, we lose APC. This protein doesn't exist anymore in the cell to destroy this yellow signaling protein. In the absence of APC, we always get stable beta-catenin. We always get transcription of these genes, and these cells continue to divide. In the absence of APC, these cells continue to divide even when the Wnt signal is not present on the surface of the cell. That is cancer. It's APC's job to make sure that gut cells don't divide when the Wnt signal is absent. But when APC alleles are mutated or lost, the gut cells do divide in the absence of the Wnt signal, causing cancer. Lose APC, and the gut cells divide too much, causing cancer to develop. So how can we treat this horrible disease? Because all new cancer types represent a different set of specific mutations, and cancerous cells continue to mutate in real time, it's likely never going to be possible to develop one universal cure for all cancer. Cancer tumors are too varied and too different from one another. But better treatments are always on the way. Better treatments are always years away from now. And as we understand cancer more and more, we can continue to understand how cancer cells are broken, understand how cancer cells are different from our own healthy cells, and exploit those differences and make those repairs. We can begin to devise treatment strategies that specifically target the cancer cells and leave our own healthy cells unharmed. One key, of course, to better cancer patient treatment is early detection. The sooner a tumor is detected, the smaller it is. We knew that, I think, already before this, the more easily removed it is. But now what we can appreciate after this lecture is the sooner a tumor is detected, the less time those tumor cells have had to mutate, the less time those tumor cells have had to get even more cancerous, to become even more asocial, to become even more selfish, and the easier they are to corral and control. But we started using cancer's advantages against it with some success. Cancer, to be successful, needs to be rapidly dividing. But the cost of that rapid cell division is limited to no DNA repair because the cell cycle never stops and the cell cycle checkpoints never kick in. I'm sure you've heard of radiation treatment for treating cancer. Radiation treatment works by bombarding the tumor with so much DNA damage that the cells die as a result of that damage. Neighboring healthy cells have the time to stop the cell cycle and repair the DNA damage that was wrought by that radiation treatment. So the radiation treatment does damage all cells, but because these cancer cells are blowing through the cell cycle so quickly, never trying to fix their DNA damage, the cancer cells die as a result. Again, exploiting that weakness. Our own cells have capabilities to fix that damage. A more recent drug works by inhibiting a more primitive DNA repair pathway that all of our cells have. Cancer cells use this primitive DNA repair pathway because it doesn't rely on a cell cycle arrest, but our other healthy cells avoid it because it's not very good. It's very primitive. Our other cells can halt the cell cycle and repair DNA more efficiently. The drug doesn't affect healthy cells because healthy cells don't use this primitive DNA repair pathway, but since cancer cells are using it almost exclusively, the drug affects cancer cells. In the presence of this drug, cancer cells have no way to repair their DNA. They're moving much too quickly through the cell cycle to repair their DNA traditionally, and the drug has taken away the primitive pathway, and so these cells are extremely susceptible to DNA damage. We're seeing very good treatment effects with lower doses of radiation in combination with this drug having cell death in cancer cells. Other modern strategies involve blocking the formation of those new blood vessels, which would feed the tumor. This doesn't keep the tumor from developing, but it keeps the tumor from growing and keeps the cells localized. It starves the tumor for the nutrients it would need to fuel that rapid cell division, impeding the tumor growth. Vaccines are also being developed, which would pre-program your immune system to recognize unique proteins that are only on the surface of cancer cells. You would essentially have your immune system cells fight the cancer cells, the way that it would fight a foreign pathogen, if we can train the immune system to see cancer cells as foreign. More targeted approaches for specific cancers involve affecting specific cancer-causing physiology, so we can block the cell signaling cascade that leads to cell proliferation. A perfect example is colorectal cancer. If we can find a way to get functional APC protein back into those cancerous cells, we would stop the cancer. We would block proliferative cell growth. Another example is here. There's a gene called Gleevec. A drug called Gleevec. This is to treat chronic myeloid leukemia, or CML. Here we have a protein that binds to ATP. It uses that ATP to phosphorylate its substrate, or its cognate protein. Once phosphorylated, this substrate protein becomes active and promotes cancer through an effector molecule. If we can block this phosphorylation, we block the activation of the effector, we block the leukemia. This drug, Gleevec, it binds to this ATP binding pocket in this protein. It blocks that pocket. ATP can no longer bind here. There's no more source for the phosphate group. We block the activation of the substrate. We block the effector molecule. We stop the cancer. Cancer is awful. Cancer is deadly. And cancer is not going anywhere. But good cell biology lets us understand how cells should be working. It lays the groundwork for fixing a broken cell. And within this framework, within this understanding, the future remains bright for the fight against cancer. Keep in mind that the study of cell biology is our best defense against cancer. But ironically, cancer has been our most informative tool for understanding cell biology. So what did we talk about in this lecture? Our bodies have this wonderful mechanism for repairing and renewing, and without this mechanism, our lifetime would be limited by the lifetime of our individual cells. But there is a price for having a pool of replenishing cells, a pool of cells that's poised for rapid division. When things go wrong, and these cells cannot be controlled, they go wrong catastrophically and cancer can develop. Cancer cells are defined by two main properties, which they pass down to their daughter cells. They proliferate like mad, and they are invasive. This is cancer. Metastatic tumors are particularly dangerous, and this is when malignant invasive cells have invaded the circulatory system, and they can spread to different parts of the body. Cancer rarely results from a single mutation. Instead, it's a disease of accumulated mutations that accumulate over time. A very specific set of mutations must accumulate together in a single cell in order for cancer to develop. When mutations affect the accurate replication and maintenance of the genome, we see genetic instability. Genetic instability leads to large-scale mutations, often at the chromosomal level, and this greatly enhances the accumulations of mutations that can cause cancer. Cancer cells no longer require signals from other cells to thrive. This makes them deaf to the control of the larger cellular communities. Cancer cells do not default into apoptosis. They will not self-sacrifice for the greater health of the organism. Cancer cells are immortal and never stop cycling through the cell cycle. Cancer cells are genetically unstable, and as a result pick up chromosomal mutations at a much higher rate than normal healthy cells. Cancer cells are abnormally invasive. They can adhere to, survive at and in, and divide at and in foreign tissues. They can also lack adhesion properties, allowing them to migrate. We defined and discussed oncogenes and tumor suppressor genes. Tumor suppressor genes giving rise to genetic predispositions because tumor suppressor gene mutations are recessive. And finally, we used colorectal cancer as an example for some specifics regarding cancer, and we discussed some strategies for future therapies of cancer, knowing what we know now about how cancer develops. This is the end of cell and molecular biology. Thank you for a wonderful semester. We're not done yet, of course. We have a number of exams to get through, but there will be no more new information. This is the end of the course with regards to lecture content, and it was my pleasure. Thanks for watching this lecture and all the others that came before it, and I will see you all in class.